Hello guys, so this is Dr. Krishna and welcome back. Today we shall be discussing the introduction part to the bacteriology, right? So you have heard many things like gram-positive bacteria, gram-negative bacteria. You have also heard about some organisms are Urease positive, some are Catalase positive, right? So some are aerobic, some are anaerobic. So we shall be discussing what is the concept behind them and we shall discuss everything in detail, guys. So this will be not only the introduction, this will be the basic class, right? This will form your basics for the entire part of your bacteriology topics, right? So starting on with, let us discuss from the basic. As I already told you, let us discuss from the basic. Let us discuss the structure of the bacteria. How does a bacteria basically look, right? So as you can see here, this thing which I'm drawing over here, this is your bacteria, right? So whatever is located within this is called as cytoplasm. So surrounding the cytoplasm, this dark black line which you can see, this is called as your plasma membrane. So let me draw the things and then later on I'll mention about everything, right? So surrounding this, we have got a very important thing which I'm drawing it in blue. So this is called as your capsule. Surrounding the capsule, you have got, what is this? This is called as your cell wall of the bacteria. This is called as your cell wall of the bacteria, right? Next, you know one very important thing, that coming from this capsule, you have got very small things that are coming out from this capsule, small projections like this, right? At the same time, you have also got some long projections that are coming out of this capsule. So these small projections are called as fimbriae or pili. These long projections are called as flagella, obviously, and with the help of the flagella only the bacteria will be moved. So when you are studying bacteriology, when you are studying different types of bacteria, when you ask the properties of the bacteria, right? Properties will be like coming to the structure of the bacteria. The structure morphology will be like some are aflagellated, some are flagellated, right? So these things you'll be hearing again and again. So bacteria which have flagella are flagellated, obviously. So let me write down the things. So this one over here, this is your plasma membrane, right? So this will be your plasma membrane, right? Next, this entire thing which I'm drawing right now like this, which I'm highlighting it exactly. So this will be your capsule. I'll tell you what is the important thing about capsule. And next, this part is called as your cell wall, right? So some organisms have cell wall, some organisms do not have cell wall. Same way, this pink color things which are look here is fimbriae. Fimbriae are also called as pili. And finally, these are called as flagella. So we shall be discussing about each and everything right now. Okay, right. Now, one important thing is that these flagella, cell wall, capsule, fimbriae or pili, what are they consisting of? I mean, what are they made up of and what is their function? Why should we discuss about them? Why should we discuss about them is that, I hope you understood this thing. Now let me go on to the next thing. Next thing is that, for example, if we make out a structure, we write on the side heading as structure and let us say chemical composition, and let us talk about the function, right? So the first structure which we shall be discussing is flagella. What is the best thing which you can tell about flagella? The best thing which you can tell about flagella is that, what is flagella made up of? Flagella is made up of proteins, right? So flagella is made up of proteins, right? What is the function of flagella? Motility. But is this enough? These are the things which are going to ask you in the exam? No. What they're going to ask you in the exam is that, look here, as I already told you, this is your bacteria over here, right? So coming out of the bacteria like this, you have got your flagella, right? This is your flagella that is coming out of the bacteria. Now, exactly if you look microscopically into the flagella, within this flagella there is a small protein which is pink in color. I mean, I drew it as pink in color, but there is a small protein which in that flagella. Now this protein is named as flagellin. This protein is named as flagellin, right? This protein is named as flagellin, or it is also called as flagellar antigen. Flagellar antigen, or it is also called as H antigen, right? So this protein is called flagellin, or also called as flagellar antigen or H antigen. What is the specialty about this? The specialty about this is it is heat liable. The specialty about this is heat liable. This is heat liable. Now we have discussed about flagella. Now let us discuss about the small things, small projections which are coming out of the capsule. These small projections are called as pili or fimbriae, right? So these small projections are called pili or fimbriae. So if I ask you what is this so-called pili or fimbriae made up of? Pili and fimbriae are made up of a protein called as glycoprotein. They are made up of glycoprotein, right? So what is the use of this pili or fimbriae? Are they also like flagella useful for motility? No. For example, this is my hand. This is the place where if the bacteria wants to stick. So what will happen is that with the help of pili or fimbriae, this bacteria comes and attaches to the surface of the host. I am the host. So this bacteria attaches to me. So for adherence of the bacteria. So what is the function? Function is for the adherence of the bacteria to the host. Adherence of the bacteria to the host, right? Next important thing is there are some bacteria, guys, very important. There are some bacteria which releases spores. These are called spore forming bacteria. For example, there are spore forming bacteria like Clostridia, right? You have got Clostridium tetani, Clostridium botulinum, Clostridium perfringens, Clostridium difficile. So all of these Clostridial species, they release spores. But whereas other bacteria like Staphylococcus, Streptococcus, they do not release spores. Now, why I am so fond of spores? Because these spores are very toxic, guys. You know, let me give an example. For example, in Clostridium tetani, the spores which are released, right? These spores, once a neonate, right? Immediately after a newborn or a neonate, when you are feeding the neonate with the honey is colonized or contaminated with Clostridium tetani spores. So the moment you take the honey, the moment you feed the baby with the honey, raw honey, the spores also enter inside. The spores will directly go to the colon, and in the colon the spores will germinate, the spores will divide, and the spores will release a toxin called as exotoxin. And this exotoxin will cause neonatal tetanus. Okay, it will cause neonatal tetanus. So that is very important thing which you need to keep in your mind, right? So why I'm so fond of these spores, guys? The reason is that some bacteria release spores, some bacteria do not release spores. Okay, so bacteria which release spores are called as spore forming bacteria. For example, the Clostridium species, bacteria which do not release spores are non-spore forming bacteria. For example, let us say Streptococcus species and all the Staphylococcus species, right? So when you come to the Clostridium species like Clostridium tetani, Clostridium difficile, Clostridium botulinum, Clostridium perfringens, so all these 4 different types of Clostridia, they release spores, right? They release spores. Why I'm so fond of spores? The reason is, for example, let us take an example of, okay, let me put a side heading, spores. Let us take an example of Clostridium, okay? Clostridium botulinum. Now, this Clostridium botulinum releases spores, right? So, a newborn cannot eat rice or chicken, right? So, in newborn, you feed the newborn with the raw honey. You mix it in the milk or in the cereals or whatever it is, but you feed the newborn with the raw honey. So, when you are feeding the newborn with the raw honey is said to be contaminated with these spores of Clostridium botulinum. So, these spores in that raw honey when the baby ingests it, when you're feeding the baby, when you're feeding the neonate, these spores, they enter into the body of the neonate. Where it will go? It will directly go and lodge in the large intestine. So these spores will enter into the large intestine of neonate. So what will happen? What will happen is that these spores in the large intestine, they germinate and they release a potent toxin called as exotoxin. I have explained this clearly in a Clostridium tetani lecture separately. So you will have very detailed explanation there, but this is just an introduction. So they will release a important toxin called exotoxin. And what is this exotoxin going to do? This exotoxin will cause neonatal botulism or infantile botulism. This will cause infantile botulism. So that is the reason why these spores are very important. And some of the spores like Clostridium tetani spores and different types of spores, they are even neurotoxic. They can even damage your nervous system. Okay, fine. So anyways, let me discuss some important thing about these spores. First thing is that bacteria which release spores are called spore forming bacteria. Let me write down the example over here as well. Best example is Clostridium SPS, which means species. Okay, so Clostridium species. Fine. First important point about the spores. The first important point is, where do you find the spores? Spores are only and only found in gram-positive bacteria. Gram-positive only. Point number 2. What is the important point number 2? Right. Are these spores heat stable? Obviously, yes. These spores are heat stable. Spores are heat stable. Now you can ask me, spore is very minute thing. I even sometimes can't see with my naked eye. How do you think they are heat stable? How do you think they survive even at high temperatures? The reason is, guys, if you look at the spore, let us say this is the structure of the spore. Okay. Now within the spore, deep inside the spore, you have got this blue color dots, right? These blue color dots are nothing but and very important acid called dipicolinic acid. With the help of this dipicolinic acid, these spores are becoming heat stable. What they contain? They contain dipicolinic acid. Dipicolinic acid makes these spores stable. Okay, fine. So can I tell that this can be an MCQ? Obviously, yes, this will be an MCQ, obviously. This is an important thing. It can be an MCQ. Point number 3. Right. So what is the point number 3? Not only heat stable, not chemically stable, obviously. It is also chemically stable. Right. Now, during dehydrated states, we basically die. Right. We get sunken eyes, there will be loss of turgor in our skin and we die. So during dehydrated states, does these bacteria survive? Does these spores survive? Yes. Even these spores survive in dehydrated state. They survive. So these are 4 characteristics of the spores, right? So regarding the spores, we shall have a very detailed lecture later on. So this is something basic I wanted to discuss in the first part regarding the structure of the bacteria, right? Now, what are the important things to cover here? The important things are this thing, flagellin makes an MCQ. This will be an MCQ, and pili are formed by glycoprotein, right? Pili are formed by glycoprotein. So glycoprotein will make an MCQ for you. And regarding the spores, you know the dipicolinic acid and what are the properties? 4 different properties of these spores. Cool. Now let us enter into the famous topic which every one of you know, the differences between gram positive and gram negative bacteria. So what are gram positive and gram negative bacteria? The real difference between gram positive and gram negative is, you can look at the picture itself. This thick blue color thing which I have drawn here, right? This is nothing but called as peptidoglycan layer, right? So this is called as your peptidoglycan layer. So the same as here, this is also a peptidoglycan layer. Look where the peptidoglycan layer is thick. Obviously peptidoglycan layer is thick in gram positive ones, right? So can I write regarding the thickness, if you ask me a question, can I tell that peptidoglycan layer is thick in gram positive and peptidoglycan layer is thin in gram negative? Yes. Next important thing, below that peptidoglycan layer you can see some black sticks with green color heads over there, green color circles. This is nothing but called as phospholipid layer, and there are two. So can I call phospholipid bilayer? Yes, this is called phospholipid bilayer. Look to the gram negative one. Even in the gram negative bacteria, you have got phospholipid bilayer. Whereas in this phospholipid bilayer is called as outer membrane, and this phospholipid bilayer is called as plasma membrane. One is called outer membrane, another one is called as plasma membrane. Between that, sandwiched between the outer membrane and plasma membrane, I have got peptidoglycan layer. Cool. Next, if I ask about peptidoglycan layer, where it is thick, where it is thin. Obviously, here it is thick and here it is thin. Right? Next important thing is teichoic acid. So what is important thing about teichoic acid? I'm telling you guys, teichoic acid is most commonly present in gram positive bacteria. What is the use of this teichoic acid is that teichoic acid maintains the structural integrity of the bacteria. It maintains the shape of the bacteria, right? This thing every one of you know. But what is that important thing which you need to know? Apart from maintaining the structural integrity, teichoic acid also is responsible for the release of two cytokines. One is called as interferon alpha and interleukin 1. Okay, so teichoic acid is responsible for synthesis of interferon alpha and interleukin 1. So it is responsible for synthesis of interferon alpha and interleukin 1. Why are you so highlighting this? What is the use of release of interferon alpha and interleukin 1? I'll tell you later on when I'm discussing exotoxins and endotoxins. Okay. For now, you need to know bacteria which have teichoic acid are gram positive, bacteria which do not have teichoic acid are gram negative. And they are responsible for structural integrity and they release these two cytokines. Next, let us go to amino acids. Do all the bacteria release amino acids? Yes, all the bacteria release amino acid. But the important thing is gram positive releases only few amino acids, whereas gram negative synthesizes or has many amino acids. Next important thing. After that, the lipid content. Where is the lipid content present? The lipid content is present in gram negative bacteria and absent in gram positive bacteria. Okay. Next, porin proteins. What are porin proteins? You know, every cell in our body have got some porins. Porins means the ion gated channels, right? Sodium ion channels, potassium ion channels, calcium ion channels. From where the sodium comes in, potassium goes out, right? Glucose transporters. So all these transporter proteins, all these gated channels, all these are pores in the bacteria, right? Pores in our own cells as well, with the help of which there is diffusion of electrolytes inside and outside the cell, right? You need some channels, right? Those channels are called as porin proteins. And where do you find those channels? These channels are found, let me write it here, these are transporter proteins. Okay, transporter. I hope you know symport, antiport, all of you know it, right? So where do you find them? You find them in case of gram positive but absent in gram positive. Next, periplasmic region. What do you mean by periplasmic region? Where is periplasmic region present? Look at the picture above. You have got something called outer membrane here. Yes or no? You have got outer membrane, right? So this is your outer membrane, and you have got plasma membrane below. So between the outer membrane and plasma membrane, whatever place is located there, that place is called as periplasmic region. This is called as periplasmic region. If I ask you a simple question, what are the contents of periplasmic region? So obviously, what will be the content of periplasmic region? The content of periplasmic region will be peptidoglycan. And where do you find periplasmic? Do I find on gram positive? No, I don't find there. So can I write that I don't find that periplasmic in gram positive, but I'll find it in gram negative bacteria. So these are some of the basic differences which everyone has to know about gram positive and gram negative. I hope I explained it with the concept. Fine. So I hope you understood it. So let us move on, guys. Let us move on. Second important thing is that now these things which I'm going to discuss, each one can be an MCQ for you. Fine. We discussed there are different types of structures surrounding the bacteria. One structure, name me please. That was your capsule, right? Capsule is one of the structure. Next, after the capsule, we discussed something called as glycocalyx. I'll tell you what is glycocalyx. Next, we discussed periplasmic region. Periplasmic region. Alright. So these three things are very important for you. They can ask a question in the exam. Now, what is the important thing which you need to know here? The important thing is, what is the function of capsule? What is the function of capsule, glycocalyx and periplasmic region? What is the function of capsule is that you have seen the capsule, right? Surrounding the bacteria, there is a capsule from where fimbriae, pili and flagella were coming out. So this capsule function is, if this is a bacteria, surrounding this bacteria, there is a capsule, let us say, right? For example, this is a phagocyte. Let us say this small thing is a phagocyte, and this phagocyte is coming all the way and it looks at it and thinks it is a bacteria. Now this phagocyte will move in this direction to kill the bacteria, but this capsule which is surrounding the bacteria will not let the phagocyte attack. It will repel away the phagocyte. So it will prevent the phagocyte to attack him. This is called prevention of phagocytosis. So the function of the capsule is that phagocytosis prevention. It prevents phagocytosis. What is the function of glycocalyx, guys? What is the function of glycocalyx is that, look here guys, surrounding the bacteria, you have got a layer called glycocalyx. Glycocalyx is like a jelly layer. Let me write it down. Glycocalyx is a jelly-like matrix. Glycocalyx is a jelly-like matrix. What is the important thing I have to tell about glycocalyx is that, for example, let us say this is a bacteria over here. Surrounding this bacteria, there is glycocalyx, which is like a gum, which is like a gel, right? Now, whenever you are inserting a catheter, and this bacteria are most commonly present on your skin. Let us take an example of a bacteria called Staphylococcus epidermidis. Staphylococcus epidermidis. Now this Staphylococcus epidermidis is normally present on my skin, right? It is a normal flora on my skin. Now when you are trying to insert a cannula, when you are trying to insert a needle into my skin, what will happen is this Staphylococcus epidermidis, it is having glycocalyx, so it will very easily jump onto that needle and it will stick to that needle, stick to that catheter, stick to that device, hospital devices, any device it might be. And how is it sticking so firmly? Because I told you the bacteria is having a glycocalyx, which is a sticky layer, which is a jelly layer. So it can very easily stick to that, very easily stick to that hospital acquired devices. So obviously when you are inserting it inside, along with that, the bacteria also goes inside. So can I tell you that if you are using catheters, or if you are using any kind of hospital devices, hospital devices, all the invasive methods I'm talking about, catheters or any kind of hospital acquired devices. So with the help of these devices also, glycocalyx containing bacteria is entering into my body. Yes or no? Right? So even through hospitals also we get infections. Right? So what was that example which I gave you guys? The example which I gave you was Staphylococcus epidermidis. Staphylococcus epidermidis. Fine. Coming to the periplasmic region. You know, we discussed about periplasmic region. But tell me, what is periplasmic region? Why is it so important? Why is it so important is periplasmic region is having hydrolytic enzymes. Periplasmic region is having hydrolytic enzymes. Can you name me one? Yes. One important hydrolytic enzyme is beta lactamase. Beta lactamase is one hydrolytic enzyme. Cool. So these three things, guys, which I've discussed just now, right? These three things will form an MCQ for you, and this is very, very, very important. You just have to memorize these things. Okay. Now, let us discuss another important thing. Now, the next important thing is that 1, 2, 3, and 4. We have discussed something called as capsule. We have discussed something called as cell wall, right? We have also discussed something called flagella, and we have also discussed short projections called as, very good, fimbriae, right? What type of antigen is located in all of these? What type of antigen do you find in all of this? One thing all of you know, flagella has got a protein called flagellin, or also called as antigen what? Antigen H. How do I remember? How do I remember is two L's, put a gap line in between, this will become H antigen. Capsule. Capsule. How does it sound? Capsule. K. So it has got K antigen. Right? Cell wall. You know, wall is so hard to hit. The moment you hit the wall, it is O. Oh my god, it's wall. So it has got O antigen. And in the same way, fimbriae has got many eyes in it, so it has got I antigen. You see fimbriae, here you have an I, here you have an I. Fine. So all these are the antigens which are located within. Antigens which are located within this. Clear? This is very, very important, guys. Very, very, very important thing. Another important thing which you have to discuss here is, we told you bacteria has got a capsule, right? So those bacteria which have capsule are called capsulated organisms. So what are those capsulated organisms? They can ask you in an exam. What are capsulated organisms? And what is the function of a capsule? I told you previously, the function of a capsule is it prevents phagocytosis. So capsulated organisms. So what are the different types of capsulated organisms? One is Pseudomonas aeruginosa. Next one is Streptococcus pneumoniae. Third one is the influenza. Influenza is caused by Haemophilus influenzae. Next, meningitis is caused by Neisseria meningitidis. And finally, E. coli. So all these bacteria have got capsule. So I call them as encapsulated organisms or capsulated organisms. How do I remember this? How do I remember this is that with a mnemonic, that is, please shine. What is P here? Pseudomonas aeruginosa. What is S here? Streptococcus pneumonia. What are H and I? Haemophilus influenzae. What is N? Neisseria. And what is E? That is E. coli. So these are the organisms which are capsulated. Fine. You told me that Streptococcus pneumonia is a capsulated. Haemophilus influenza is a capsulated. How am I going to prove? How am I going to prove is that I will take an agar plate. a round color agar plate which you see in your micro labs, right? So in that, I will add one of these bacteria. After adding bacteria, let us say I have added H influenza. After adding this bacteria, right? Let us say I have added Streptococcus pneumonia, right? This is simple. So I have added Streptococcus pneumonia bacteria. Now onto this, I am adding a serum. Serum contains what? Antibodies. So the moment when I add serum, what is going to happen? The antibodies within that serum, what will it do? If there is a cell wall, then they will go and attach to the cell wall of Streptococcus pneumonia. If Streptococcus pneumonia do not have any kind of capsule, then antibodies will not attach to the capsule. If that bacteria has a capsule, then it will attach to that capsule and cause swelling of that capsule. It will cause swelling of that capsule. So such reaction is called as Quilling reaction. Do I need to know about Quilling reaction? Absolutely no. What you need to know about Quilling reaction? That how do you detect a capsule? So capsule is detected by which reaction? That is Quilling reaction. What is Quilling reaction? Simple thing is that organisms which have capsule, they swell. So if you see swelling, that is positive Quilling reaction. Clear? Now after the capsule, we have discussed about the capsule part. Now let us go on to the cell wall part. Let us go on to the cell wall part. Cell wall part. You know, we have got gram positive organisms, we have got gram negative organisms, and we have also got mycobacterium. You know, mycobacterium is responsible for causing TB, right? Mycobacterium tuberculosis. So now tell me, what is the composition of cell wall of gram positive, gram negative, and mycobacterium? What I should answer now, I'm asking you, what is the composition of gram positive cell wall? Gram positive, look at the picture before. Gram positive has got a phospholipid bilayer. It has also got a peptidoglycan layer. Very good. And what does it has got is that it has got peptidoglycan layer. You know it, very thick, right? Next, along with that, it has also got teichoic acid. Next thing, gram negative bacteria. What does it got? Gram negative bacteria also has got peptidoglycan layer and not teichoic acid. I told you in gram negative, teichoic acid is absent. So it has got a peptidoglycan layer. Along with that, it has got LPS. LPS stands for lipopolysaccharide. LPS stands for lipopolysaccharide. What is mycobacterium consisting of? Mycobacterium is having again peptidoglycan. So I think peptidoglycan is basic for everything, right? Peptidoglycan plus it has also got mycolic acid. Mycolic acid. Next, it has also got a third component, that is lipoarbinomannan. Right? Now, bacteria which have mycolic acid, they are called as acid fast organisms. Bacteria which do not have mycolic acid are called acid fast, non-acid fast organism. Right? You know the geelness and staining and all, right? Anyways, we shall be discussing in later on lectures. But now what you need to know is the simple thing. They can be an MCQ. Again, I'm telling you, mind you, this can be an MCQ. Mycobacterium tuberculosis has got mycolic acid and lipoarbomannan. Right? Now, we have discussed enough. Now, the last part will be that organisms, microorganisms are divided based upon urease positive, organisms which release urease are called urease positive, urease negative. Next, organisms are divided based upon the catalase enzyme, catalase positivity. Organisms are divided based upon hemolysis. and organisms are divided based upon pigment production. So in our today's lecture, we shall be discussing organisms that are classified based upon urease and catalase. Fine. We shall discuss about hemolysis and pigment later on in next class. So based upon urease and catalase, what is a concept? Listen very carefully. Urease sensitivity, wrong. Urease test. Based upon the urease test. Now look here. One very important thing is that all of you should know that if a bacteria is releasing an enzyme urease, I call it as urease positive. If a bacteria is not releasing urease, I call it as urease negative. Why? What will happen when urease is released? Why urease should be released? Question number 1. Question number 2 is that there are bacteria in the female genital tract near the reproductive system, near the vagina, near the cervix, near the lips. Obviously, you will have a lot of bacteria. But still, why? That environment in the female vaginal tract, right? That reproductive tract, it is highly acidic because all the time the urine will be there, the secretions will be there. So it will be highly acidic all the time. So in that acidic environment, how the organisms are living? How can an organism survive in such acidic environment? The reason is those organisms which are surviving there are urease positive organisms. How are you telling me? How am I telling you is that now concentrate on this topic here. Let us see an example of right. So this is a female genital tract, right? So this is a simple picture of a female genital tract. Now in this female genital tract, let us say that you have got a bacteria over here, right? So this is the bacteria. Now this bacteria, I tell you that this bacteria releases an enzyme called as urease. So what will happen is that this urease, in the female genital tract, I told you all the time the environment is acidic. Normally bacteria cannot live there. But how the bacteria living there are, in the female genital tract, you have got a compound called urea. So what is this urease doing? Urease is converting urea into ammonia plus carbon dioxide. And what is this ammonia? Ammonia is acid or alkali? Ammonia is alkali. So more the bacteria, more the release of urease. More the release of urease, more the release of ammonia. It means the entire female reproductive tract is becoming alkaline in nature. So in this alkaline nature, obviously the bacteria can very happily survive. And this is how urease positive organisms are surviving in the female genital tract. Right? What are urease positive organisms? Let us say Proteus. Proteus is a urease positive organism. Not only Proteus, Cryptococcus. Cryptococcus is also a urease positive organism. Next thing is Ureaplasma. Ureaplasma, Nocardia, Klebsiella, and finally Staphylococcus epidermidis, which I told you. Epidermidis. So all these bacteria are releasing urease and they can live very happily in the female reproductive tract. One very important thing for your MCQ point of view is about the Proteus. What is Proteus? Proteus, this guy over here, this Proteus, what does it do is that this Proteus is also responsible for causing some stones, and these stones are made up of ammonium, magnesium phosphate. These stones are made up of ammonium, magnesium phosphate. And what is the names of these stones? The name of these stones are called as struite stones. Struide stones. This can be, again I'm telling you, this can be an MCQ. Struide stones are very important. Fine. How can I remember? How do you think I'll remember all these bacterial names? Simple thing that remember it as P chunks. P chunks. What do you mean by P chunks? P stands for Proteus, C stands for Cryptococcus, and let us say U stands for ureaplasma, N stands for nocardia, K stands for Klebsiella, and S stands for Streptococcus epidermis. Then where is H? H stands for H. pylori. That explains everything. Fine. So we are done with the urease enzymes. Now the last topic which we'll be discussing are the catalase positivity. Right? This is very important, guys. I'll tell you why. Same to same concept, organisms which release catalase are called as catalase positive organisms, and organisms which do not release catalase are called as catalase negative organisms. So what is the concept behind that? The basic thing which you need to know is that bacteria which releases catalase, what does it do is it will convert H2O2. H2O2 is hydrogen peroxide. So it will convert H2O2 into water plus half mole of oxygen. With the help of what? With the help of the enzyme called as catalase. On the other hand, H2O2 can also be converted into another compound called as OH radical. This is called as hydroxyl radical. Hydroxyl radical. You know, radicals are very powerful, guys. Normally in your body also radicals are produced, but time to time radicals are neutralized. How they are neutralized, I'll tell you in a minute. But anyways, I'm telling you, wherever you see a dot above the oxygen, above this OH or anywhere, they are called radicals. And be very careful with the radicals, because radicals can even break down your RBC. The basic thing which radicals do is they break down the RBC. They cause hemolysis. Radicals are so dangerous. Even they kill the bacteria as well. Right. So organisms which release catalase are called catalase positive. Right. So in clinical practice, how am I going to use this concept? How am I going to use this concept is that when I have wounds, when I add hydrogen peroxide, immediately I see foam. Why? Because lining within my endothelial cells, there is catalase enzyme, and this catalase enzyme will convert the hydrogen peroxide into water and oxygen. Water and oxygen together is called foam. Right? Never ever add hydrogen peroxide to deep cut wounds. For example, if I got a cut in my radial artery or femoral artery, I will not add hydrogen peroxide, because when I add hydrogen peroxide, obviously oxygen will be formed, and these oxygen bubbles, they enter into my artery and cause air embolism. So never ever add hydrogen peroxide to deep cut wounds. Right. Now, what statement I'm going to give you now is that very powerful and important statement. Patients with chronic granulomatous disease. Patients with chronic granulomatous disease, these patients will have recurrent catalase positive. So if I am having chronic granulomatous disease, I will be having recurrent catalase positive organism infections. What is chronic granulomatous disease? Those things I will be discussing later on in pathology. But now what you need to know, that patients with chronic granulomatous disease will have recurrent catalase positive organism infections. Why? The reason is, in chronic granulomatous disease, there is NADPH oxidase deficiency. There is NADPH oxidase deficiency. Fine. Can you explain it more? Yes. What is the important thing? What is the important thing is that, how can I explain this to you? Let me put a picture over here. let us say is a guy over here, right? Now, right. So let us say that there is a phagocyte. Let us say this big is the phagocyte. Phagocyte is so big here, right? This big is the phagocyte in his body. So let me enlarge it up. Okay, fine. This is a phagocyte present in the patient's body. In you and me also the phagocytes will be there normally. Now, this thing which I'm going to draw here, this green color thing, right? So this is nothing but called as lysozyme. All of you know that lysozyme has got some enzymes. These enzymes are called hydrolytic enzymes. Next, I have got another important thing. This blue color thing which I have drawn here, this blue color thing is called as phagosome. Phagocyte has got phagosome. Fine. Now, when there is a bacteria in my body, what are these phagosomes or phagocytes going to do? They will engulf the bacteria and store this bacteria within its phagosome. So should I draw few bacteria over here? See, these are few bacteria which are captured. These red color things are the bacteria which are captured. And this pink color things which I'm drawing now, this is nothing but called as your NADPH oxidase enzyme. Fine. So what are you trying to teach us? What am I trying to teach you is that normally in you and me what happens is that when you see such a situation, this lysosome, look very carefully here. This lysosome, right? Fine. So this lysosome all the way travels like this, travels and attaches with the phagosome. The moment it attaches with the phagosome, what will happen is phagosome and lysosome they join together. When they join together, that is when they form phagolysosome. Phagolysosome. So what is happening in the phagolysosome? This is your lysosome over here, and this was your phagosome over here, and you have got bacteria over here like this, and also you have got these dots called as NADPH oxidase. Listen very carefully now. This lysosome is having hydrolytic enzymes. These hydrolytic enzymes they will directly go and kill the bacteria. Second thing, these hydrolytic enzymes, they even activate NADPH oxidase. Whatever I'm telling you, let me put into words here. Lysozyme releases hydrolytic enzymes. And these hydrolytic enzymes, what are they going to do? They directly kill the bacteria. On the other hand, what is the same lysozyme doing? This lysozyme is also activating. It is also activating what? It is activating NADPH oxidase enzyme. NADPH oxidase enzyme. What will happen when NADPH oxidase is activated? Look now very carefully. Everything will come into place. Just pay attention. NADPH. This NADPH will first be converted into O2 dot, which means superoxide radical. This is a superoxide radical. And this O2 dot will further be converted into H2O2, which is hydrogen peroxide. And I told you hydrogen peroxide on one side it will be converted to water and oxygen. On the other side it will be converted to hydroxyl radical, isn't it? Hydroxyl radical. So NADP is producing superoxide. It is also responsible for producing H2O2. The process of synthesis of superoxide and H2O2 from NADP, this process is called as respiratory burst. This is called as respiratory burst. And what is the enzyme? The enzyme is NADPH oxidase. Fine. Now listen very well. Listen carefully. Lysosome activates my enzyme NADPH oxidase, and NADPH converts into superoxide. Later on, it also converts into hydroxyl radical. And where is this all happening? This is all happening right now in my body. Within that phagolysosome, within that blue color capsule here, which I represented here, within that. So within that, I have formed, very carefully look here, within that I have formed O2 dot, OH dot, both of them. And this O2 dot and OH dot, what are they going to do? They will kill down the bacteria. Bacteria is dead now. Bacteria is dead. So now you might be thinking that patients with chronic granulomatous disease, they don't have this NADPH oxidase. When they don't have this NADPH oxidase, do you think NADPH can cause respiratory burst? Do you think NADPH can release superoxide and hydroxyl radical? No. So when superoxide and hydroxyl radical are not released, do you think the bacteria within this phagosome will be damaged? Will be killed? No. When there are no free radicals, how are you thinking that the bacteria will be killed? Right at this time, right at this point, catalase positive organisms come inside. And catalase positive organisms will tell you that, hey, look, you don't have NADPH oxidase. We are catalase positive organisms and you don't have NADPH oxidase. As a result, there are no free radicals produced in your body. But we as catalase positive organisms, we have an ability to release a single radical that is called as OH dot, that is hydroxyl radical. So if you let me inside, then I'll help you to kill down the bacteria. So in this way, what as a person does is that this guy allows catalase positive organisms to enter inside. And obviously catalase positive organisms will release what? Hydroxyl radical and start killing this bacteria. But on the other hand, catalase positive organisms also cause diseases in our body. So in this way, in these patients, what is happening? You are attracting, I know you are a patient of chronic granulomatous disease with NADPH deficiency. But on the other hand, what you are doing is you are attracting these catalase positive organisms to enter into your body. So as a result, the catalase positive infections are also increasing in your body. And that is the reason why I told you that patients with chronic granulomatous disease will have recurrent catalase organism infections. Clear? Now, what organisms release catalase and what organisms do not release catalase? So organisms which release catalase are catalase positive, right? So Candida is catalase positive. Aspergillus, Nocardia. Not only that, even Pseudomonas, Listeria, you know Listeria monocytogenes, E. coli, and Staphylococci. All these bacteria are called as catalase positive organisms. They are called as catalase positive organisms, right? Fine. Now it comes to the remembering part. How am I going to remember all these things? How you are going to remember is with the mnemonic again, that is cats, catalase is CAT, cat, right? So cats need, if cat has to live, it need a place. So cats need places to live. So what does C stand for? C stands for Candida, A stands for Aspergillus, and N stands for Nocardia, P stands for Pseudomonas, L stands for Listeria, C stands for, there is no C, cool, E and S. E stands for E. coli and S stands for Staphylococci. So cats need places to live, right? So these are the basic things which we thought of discussing in this lecture. So in the next lecture, we shall be continuing with the bacteria which are what? Which are hemolytic bacteria which are pigment producing. We shall be discussing what are the different types of pigments a bacteria produces. So thank you so much.